MTT Assay slide is visible to you people?
Yes, sir.
Right. Okay, let's start then. Actually, as we discussed yesterday, that we can check the cell viability by different methods. One of them was through the hemocytometer that we discussed yesterday. Today, we will discuss the MTT assay. This assay is also used for the cell viability assessment. So here at the bottom, you can see the yellow color. This is the powder. Actually, this is how this MTT look like. We make the solution and use for cell viability testing experiment. And this, as you can see here, the powder color is yellow, but after um, chemical reaction in the cell, it converts into the purple color, the pharmazone. And this purple color you can see in the 96 well plate. So this color change actually represents the how much number of cells are alive. So if we track this change in color, we can find out the live cells. So this MTT assay is actually uh, enzymatic reaction in which the tetrazolium salt, I mean the MTT, convert into pharmazone crystal. This pharmazone crystal you can see over here. This is the MTT structure which convert into the pharmazone. In which cells? The cells which are alive, whose mitochondria is working correctly. Those cells, the metabolically active cells, they can convert this MTT to pharmazone only. The dead cell, of course, they will not be able to convert this. So this pharmazone can be solubilized, producing a color which is purple, you can see here, and we can track this change in color using absorbance like 570 nanometer, and we can use the plate reader for this purpose. If we are using the 96 well plate, we can have the plate reader which can read this plate at once. So we just need to set the plate reader at wavelength that is 570 nanometer, and it will detect this purple color, that is the pharmazone color. So the color intensity will represents the alive cell number. If we have more purple color here, then it means we have more alive cells. If we have less pharmazone purple color, which means there are less cells with which are metabolically active, mean there are less number of cells which are alive. So it is directly proportional to the metabolic activity of the live cells. So to check the cell viability using the MTT assay, first what we need to do is we need to have the cell culture. Usually whenever you want to perform the cytotoxicity experiment, you need to spare 3 days for you need to dedicate 3 days for this experiment. The first day is basically for the cell culture, and the second day is basically for the treatment of the drug whose effect you want to check, and the third day usually you spent on the mitochondrial activity testing with the help of this MTT assay. So in this way you need 3 days. So whenever you design such cytotoxic experiment, you need to uh, schedule 3 days. You need to first of all you need to culture the cells, which means you should have enough cells in your culture flasks. If you have enough cells in culture flask, then you will take the flask, uh, and of course you are doing this whole cell experiment in the clean bench. You cannot do outside because of the chances of contamination. That contamination could be microbial or something else. So to avoid that contamination, you must need a good uh, clean bench that we already have in our lab, zoological lab. The laminar flow you might have seen, which have the glass in front, where the experiment person researcher is sitting. So that laminar flow you must need for all the cell experiments. That will help you to reduce the chances of contamination. So assume you have the cell culture flask which is on the laminar flow table. Then you will discard the media and wash the cells with PBS, and then you will put the trypsin to detach the cells, as you can see here in the first line. So this is what I am trying to explain. When you detach the cells, then you will collect them with the help of electric pipette to the tube that might be 15 ml conical tube. You will centrifuge and you will get the cell pellet at the bottom. The upper media, of course, which is called the supernatant, that will contain the trypsin as well. You cannot use that trypsinized media anymore. Then you will discard this media. Only you will have the pellet at the bottom of that tube. And here you need to do the next step very quickly, that is the addition of the fresh media. Because if you will not introduce the media quickly, then what will happen? The cell can dry and they can die. So the mortality process can start. To avoid this, you must need to do these all steps very quickly. Now the cell pellet is mixed with the fresh media and you have the cell suspension. This cell suspension you will use for your experiment. You will take the well plate. We have 6 well plate, 12 well plate, 24 well plate, 48, 96. These are the well plates which you usually use in the cell experiments. So for this purpose, for cytotoxicity testing, you can use anyone, but mostly people prefer 24 because of the enough number of cell, enough space for the uh, cultured media and the cells to grow. So let's suppose we are using 24 well plate. The cell suspension that you already have, you will take 1 ml cell suspension and put in uh, all the 24 wells. If you are uh, using some drugs which you want to use in dilution and want to check the dilution effect, you can uh, fill the cells with the cells, fill the wells with the cells as much as you want. So you can design, if you have just 2 drugs, maybe you can use 2 wells, or the the maybe 9 wells in if you want to use the same concentration in triplet. So in this way you will get 3 wells for each cell dilute for the drug concentration. So here assume we have 24 well plate. You are using first column only 3 wells, in second column only 3 wells, third column you are using only 3. So in total in 3 column you have 9 wells with cell. This, if when you put the cell inside the well and with the media, this is called basically the seeding of cell in the well plate. So when you are done with the cell seeding, you will incubate this plate and let the cells to attach with the bottom of that plate. So usually we incubate for 24 hours. Here on the right side you can see the incubator that we usually use for incubation. This is cell CO2 incubator. After appropriate incubation, again here you must need to understand you can incubate for 12 hour, maybe 16 hour or 24 hour, and you can check the cell drug effect. And this is the second incubation after introducing the drug. And you can use any stimulus for this testing. For example, you can use the plasma and you can check its cytotoxic effect. You can use some uh, drugs, for example, drug A, drug B, drug C, whatever. So how you can do that? Here in this well plate, this is maybe 12 well plate, and the cells are seated over here. So let's assume in these 3 cells you did not expose with anything. So this is called the control. However, in this column, these 3 wells, you are exposing these cells with 1 minute plasma treatment. And in the next column here, in each well, you are treating with plasma with 5 minutes. And the last column, you are exposing the cells with 10-minute plasma treatment. So the first column you have the cell that are controlled, which means they are not treated with plasma at all. However, the second column, third column and fourth column, they are treated sample but with different dose of plasma. Like the second column is for just 3-minute treatment. The third column is exposed with plasma for 5 minutes, and the last column is exposed to the plasma for 10 minutes. And you want to check the plasma effect. For example, uh, you can take some other drugs as well, like a drug A, drug B, like doxorubicin. This is the drug we usually use for the cancer. You can check their effect as well on the cells. You can check the you can take the HeLa cells, which are the cervical cancer cells, and introduce that doxorubicin and check the killing effect. So you can use either plasma or other drugs, or whatever the chemical you are testing, the method will be almost the same. So when you introduce your drug, or when you are done with the plasma treatment, you will put this well plate to in this incubator, and you will incubate for your desired time. Usually we incubate for 24 hours and we check the 24-hour drug effect. However, you can reduce. Uh, you may check the time dependent drug effect as well. For example, you want to check the same drug effect after 4 hours, after 12 hours, after 16 hours, or after 24 hours. In that case, you need one plate for 4-hour incubation, one another plate, second plate for 12-hour incubation, third plate for 16-hour incubation, and fourth plate for 24-hour incubation. So these all plates have the same number of cells. They have all parameter same. The treatment is same. Only the difference is the incubation time. And after desired incubation, you can perform the MTT experiment. You can introduce the MTT. You need to put the MTT in each well, and you can incubate again for 2 to 4 hours. But whatever the incubation time you select this time, for example, 2-hour incubation after MTT addition, you must need to keep the same incubation for rest of your plates. For example, for 12-hour incubation plate, for 16-hour incubation plate, and 24-hour incubation plate. So when this 2-hour incubation is done, then what you need to do is you have to introduce the DMSO. First of all, you will discard the MTT and the media that you have in the well plate during 2-hour incubation, and you will add the DMSO and some buffer like Sorensen buffer, and then this DMSO and buffer will make your cells burst, and the formazone formed inside the cell will come out. Now this formazone, if you have more cell alive, it will have the more purple color intensity. If the cells are less in number, alive cells are very less in number, then this color intensity will be very low, as we explained in the MTT reaction process. So this change in color you will track at 570 nanometer. You can use the plate reader, and you can insert this 96 well plate and set your plate reader to read at 570 nanometer. It will give you digits, and you can compare that values with control sample. So if you have 100 cells in control, and for drug A or plasma treatment time 3, you get for example 90 percent intensity, it means 10% are dying. And in the next treatment, that is the higher than the first one, which is 5-minute treatment, you have maybe 50 percent color intensity as compared to the control. So you have the 50% alive, 50% dead. And in the last 10-minute plasma treatment, maybe you have the 20% purple color intensity as compared to the control. In this case, you have 80% death rate. So this way you will use this MTT assay to find out the viable cells, and at the end you will plot that values that you get from the plate reader and make the graph like this one. These graphs are also showing the cell viability of 3 cell lines. Here you can see B16F10 cell, which are the melanoma cells. These are the Hackett, which are, of course, all these cells are skin cells. And the last one is L929. These are the fibroblast cells. So these all cells were treated with different concentration of one of the chemical, that is 4B, as you can see here, and their killing effect was checked and finally was plotted with the help of MTT. This is the same method you discussed today. So this graph is taken from this scientific report paper. I will just show you quickly this paper.